latest research being conducted at Chordoma Foundation Labs. The research is essential to accelerating progress in chordoma treatment, aiming to identify and develop new therapies and improve patient outcomes. And Dan is here to share more with us about the exciting progress on that front. So welcome, Dan.
Woo! So Dan.
Hello. Okay, so it's great to be here and see some familiar faces and also lots of new faces. Really have to give a research update today, not just of the research that we've been doing in CF Labs, but I'm actually also going to talk about some of the research that we've been supporting in the community, so it'll be a little bit more high level in that sense. Okay, so I think many of you already know this, but for those of you who are kind of new to the community, the mission that we have at the foundation is to improve the lives of those affected by chordoma and lead the search for a cure. We kind of have three major thrusts, and I'm sure you've heard about many of these already today. So patient services to provide a better experience, healthcare improvement to provide better care, and research to deliver better treatments so that patients can overcome this disease and maintain their quality of life. And today I'm going to be talking about our efforts on the research front to develop better treatments. Okay, so in the research department at the Chordoma Foundation, priority number one for us is developing effective drug therapies. Okay, so not because we don't care about advances in surgery and radiation therapy; those are very important as well. But priority number one for us, our main focus is on finding or developing more effective drug therapies, and predominantly that is first and foremost for patients that have unresectable, recurrent, or metastatic disease, so so-called advanced chordoma. And the goal there is to develop therapies that can control advanced disease and meaningfully extend or normalize the duration and quality of life for those patients. So that's priority number one. And then eventually we hope to develop therapies that can be used alongside surgery and radiation, and either just before surgery or just after surgery or radiation, that can both reduce the morbidity of surgery or potentially even reduce or prevent disease recurrence. So kind of sort of an adjuvant to those approaches to kind of increase their efficacy. And then the ultimate goal would be having drug therapies that are so effective and safe enough that they could be offered as a potential alternative for certain patients instead of surgery or radiation. We do expect surgery and radiation to remain a cornerstone of chordoma management moving forward, but it might be the case that one day we have drugs that are so good that they can be offered to patients as a potential alternative. But what's important to note is that there's no silver bullet, okay? We're not going to just find one drug that can be used to beat chordoma. It's going to require an entire toolbox of drugs. So we're gonna need to come up with an armamentarium of different drug therapies that can be used to give to chordoma patients, for example, sequentially. So if a patient is on a drug and that drug no longer stops working, we wanna have a plan B, we wanna have a plan C, we wanna have a plan D, so that we can give therapies to patients, and if they no longer stop working, we can have an option coming up behind it. Secondly, personalized care is obviously going to be very important. So matching the right drug with the right patient at the right time. There might be two patients in the audience right now that have conventional chordoma, but we know that the disease biology is nonetheless much more heterogeneous than just the histological subtype of your tumor. So you might have conventional chordoma, and the person next to you might have conventional chordoma, but the underlying biology and the drug vulnerabilities of that conventional chordoma might be quite different, and so you might benefit from a different drug than the person sitting next to you. So it's going to be important to find out how we can match the right drug with the right patient at the right time. And finally, combinations are going to be necessary. As with any cancer type, we are interested in developing drug combinations that can be given to patients to both enhance the magnitude and extend the duration of the anti-tumor response. So we're going to need multiple drugs, a suite of drugs for sequential therapy, for personalized oncology, and for drug combinations. And there's multiple paths to reaching this goal, to having this toolbox of drugs. There's over the past 10 to 15 years, the main focus of our efforts has been on repurposing drugs that are already approved. So these are drugs that are already on the market for other cancer types or other diseases altogether. So there's about 3,000 drugs that are currently on the market, about 250 of which are approved for various cancer types. And so over the past 10 to 15 years, we've been focusing primarily on repurposing these approved drugs for chordoma. So does chordoma share the same vulnerabilities as other cancer types to certain drugs? And we've made some advances in that area that I'll cover in just a few slides. So that's one path. The other path is repurposing experimental drugs. This is the next generation of cancer therapies. These are cutting edge drugs that are not yet approved. They're in clinical development, they're in clinical trials, or perhaps in preclinical development. And so that's one area of focus that we are now shifting our attention to. And then the third path is to develop new drugs altogether, right? So, providing more ways to attack cancer for targets that have not yet been drugged. And so along those lines, brachiury is a major focus of our efforts on the new drug discovery front. We know that it's a defining feature of chordomas and also one of its key vulnerabilities. So we're going to talk a little bit about what we're doing to develop new drugs against brachiury. Regardless of the path that we take here, each of them will traverse key areas of research. So from resource development to target discovery, to discovery and development of new drugs, generating translational data package that can justify new clinical trials. And we really need at the foundation to invest in promoting seamless progress along each step of the path in order to bring new therapies to patients with the speed and the sense of urgency that I know you all have. And so how can we do that? We can do that through a number of different efforts. One way that we can accelerate the search for better treatments is to raise money and then fund research at top-tier academic medical centers. So this is obviously very important. This is sort of the traditional role that a nonprofit organization plays, is kind of funding research at top-tier medical centers. But we know that for chordoma and for other rare diseases, this is necessary but not sufficient. So it's very important, but it's not going to do it on its own. We need to find more ways to stimulate progress. And so one of the ways that we do that is through launching CF Labs. As many of you know, a couple of years ago, we launched our own lab that's 100% focused on chordoma. Day in and day out, we are focusing on developing new treatments for chordoma. And in the lab, we really kind of play a unique role and complement the research that is done in academia and the research and development activities that are done in industry, and really try to synergize with those efforts and sort of grease the skids for drug development in chordoma. Now we have research that occurs in so three unique pillars, right? There's in the research ecosystem, you have academic research, you have industry research, and now you have this third kind of patient organization-driven research. It is absolutely critical that regardless of where the research is happening, that we facilitate the rapid exchange of information and ideas, right? And try to stimulate collaborations where potential opportunities for synergy and complementary expertise and interests exist. So a major part of what we do is generating kind of forums for knowledge exchange and idea exchange. And so a lot of this happens behind the scenes on a daily basis. Ashley and I are constantly communicating with researchers and trying to provide them with the resources that they need, trying to connect them with other like-minded researchers where opportunities exist, and also trying to rapidly share data from the lab as soon as we can. So there's a lot of kind of informal, organic knowledge exchange that we try to promote, but we also have more formal venues. So we have a research workshop that we put on every other year. This is a picture of the last research workshop that we had in Boston in July 2023. We have another one coming up in the next year or two. We also don't just put on research conferences, but we attend them as well. So we attend all of the major cancer research conferences. And in fact, the research team has been here in San Diego all week attending the Connective Tissue Oncology Society meeting, or CTOS meeting. And we had 3 presentations from the lab this week. All of them were on Thursday. So on the far left here is, here we go. On the far left here is Nindo, and he was selected for an oral presentation on the work that we've been doing in the lab to kind of promote replication stress and DNA damage repair inhibition in chordoma. And so that talk was selected for an oral presentation. And then here is Tyler, who gave a poster presentation on our efforts to develop an assay pipeline to facilitate drug discovery campaigns against brachyury, one of the key Achilles heels in Chordoma. And then on the far right here is me giving a poster presentation on our work that we've been doing to try to understand how we can deliver precision medicine or personalized medicine with EGFR inhibitors. So how can we understand which patients are most likely to benefit from EGFR inhibitors, which patients should not get an EGFR inhibitor because they're unlikely to benefit and should instead get a different drug. And I have to just harp on the research team here a little bit, because we found out yesterday that both of our posters were selected as best in their category at the So yeah, that's, if there's any lingering doubt about the quality of research that a small nonprofit organization can do, I think this erases that. So really proud of the team for how well they did with the poster presentations and then being selected for an oral presentation. So that is kind of knowledge exchange and collaboration is absolutely important. If we are going to ever successfully repurpose a drug for Chordoma, we need to understand how we can repurpose ideas, right? How can we repurpose ideas from other cancer types, from other efforts to drug different cancer types? And a lot of that is attending other conferences and promoting the exchange of ideas. Patient participation in research is absolutely critical. So we need you, right? We need the patient community. There is actually a lot that you can do to help catalyze the search for new treatments for Chordoma. So number one, participation in clinical trials. I think you probably talked about that a little bit earlier today with Dr. Kesari and his upcoming trial. When I walked in, I think Dr. Schwab was talking about how an opportunity for everybody to participate in research. There's of course the natural history study, which may have talked about earlier today, I don't know, but signing up in the natural history study, if you haven't already done so and you're interested in doing that, please get in touch with Shannon and Andrea. And then another way that patients can participate in research is through our tumor donation program. So donating your tumor to our biobank, we've now amassed several hundred tumor specimens in our biobank that we can distribute to researchers around the world or to grantees. And in some cases, we can even use those tumors to generate important research enabling resources such as cell lines and mouse models of Chordoma, which are absolutely critical to discovering and validating new therapeutic targets and translating those ideas into new clinical trials for Chordoma. So through all of those efforts, we've come a long way together. So since the foundation has been launched, we've gone from missing all critical resources that are needed to enable research to now having over 24 cell lines, over 12 mouse models of Chordoma, several hundred biospecimens, in a growing data repository. We went from having almost no therapeutic targets that were known to having identified and validated multiple therapeutic targets that can be drugged by existing therapies. We've come from not understanding brachyury's role to understanding that it is in fact a key target in this disease, but has always been assumed to be undruggable. I'll explain what that means in a little bit. To now having multiple brachyury drug discovery programs ongoing in the lab, partnering with multiple companies to try to drug brachyury.
Can I ask a question here?
Sure.
So is this brachyury driver for Chordoma?
We know from functional genomic studies that if you delete brachyury, the cells completely stop growing. So, I don't know if driver per se is the right word, but it is a dependency. That's a great question, and we don't know the answer to that. We know that, so we need to test more cells, and having a drug, or at least a tool compound, will help that. The cells that we've tested so far stop growing. They completely stop growing, but they don't seem to die. It might vary from cell to cell to cell.
Is this like Chordoma cell line?
Yes. And we also don't know what happens in the context of a tumor. Right, so
can you do like a 3D culture to make sure it's like more mimic the legal environment?
Yeah, we're working on 3D cultures, and we also have PDX models, mouse models of Chordoma. The problem is we don't have a drug, but we could explore if we can achieve potent knockdown in the context of a 3D model, we could do that. I'm a little bit concerned about how easy it would be to knock that down, but we have inducible, we've recently generated inducible knockdown cell lines in the lab. The problem is even in 2D, that shRNA knockdown is not fully, it's not 100%, you know, and so but that could give us a window. That could give us a window. Was there another question? Okay. So we've come from an era where no drugs were tested against chordoma to now having tested in our lab alone over 100 drugs against chordoma. Others have screened large libraries of drugs, of hundreds or thousands of drugs. So we have screened, basically, we've mined all of these repurposing opportunities, and we've identified 10 drugs out of that have significant activity in mouse models of chordoma and have been kind of whisked into clinical trials. And we've come from having one clinical trial conducted before the foundation was launched to now having supported 11 clinical trials with 5 more in planning. So lots of progress. And so there's multiple paths. There's repurposing approved drugs, repurposing experimental drugs, which are in clinical trials, and developing new drugs. I want to focus on some of the recent successes in repurposing of approved drugs for chordoma for just a moment. So here's a graph. On the y-axis is basically the percentage of patients that experience significant shrinkage of their tumor on therapy, and the x-axis is year. So here's when CF was launched. You can see there's one clinical trial that was run before Chordoma Foundation was launched, and then a whole handful of them that were subsequently run, which is great, but the problem is basically less than 5% of patients are experiencing significant tumor shrinkage. However, more recently, within the past couple of years, we have these 3 drugs, pemetrexed, pembrolizumab, and afatinib, which have produced between 10 to 15% tumor shrinkage in about 10 to 15% of cases. And so this is something that, you know, we should be really excited about and happy that we have. We have now 3 good drugs for chordoma patients: afatinib, pemetrexed, and pembrolizumab. And in the near term, what we must do as soon as possible is to make sure that we can update the treatment guidelines to enable access and insurance coverage to these drugs for as many patients as possible. So that's one of our top priorities right now. So in the bigger picture, what this tells us is that it's proof of concept that preclinical results, the results that we've generated in the lab, can translate to patients. So for example, the afatinib clinical trial was launched based on data that we generated in the lab. We found that afatinib was very active in mouse models of chordoma, and that translated into patients. So that kind of provides proof of concept that moving forward, the results that we generate in the lab can give us a pretty good sense of what we're going to see in clinical trials. Number 2, it provides anchors or opportunities for combination therapy approaches. So you heard from Dr. Kesri earlier today. He's kind of building on his results with pemetrexed that were recently published, and now launching a clinical trial with pemetrexed in combination with pembrolizumab. So combining those 2 therapies that individually worked pretty well in chordoma patients, now can they work much better in combination? There's rationale from lung cancer that these 2 drugs can work very well in combination with each other. So is that going to pan out in chordoma? We'll see. The hope is that it will. It can also provide opportunities for sequential therapy approaches as well, right? As I discussed earlier. Number 3, it highlights the importance of supporting research to enable personalized treatment. So matching the right drug with the right patient at the right time. And we're investing in a number of projects right now. We're supporting a number of grants at institutions that are trying to do that. We also have, as I alluded to earlier, a project ongoing in the lab right now where we're trying to understand the factors, the molecular factors that predict response to EGFR inhibitors in chordoma. And it marks a turning point in chordoma therapy development. So we have now mined almost all of the repurposing concepts for drugs that are approved. And we have 3 drugs that are active in chordoma patients. And that's something we should certainly celebrate, but we shouldn't settle for it either, because it's not good enough, right? We need to do better. We need to find more drugs. We need to increase the toolbox We need to find better drugs that can lead to more significant benefit in a greater percentage of the chordoma patient population. So we're at a turning point right now, and it's a completely different ballgame. So in terms of the economics and the risk and the needs of the companies that we need to partner with to get access to these experimental drugs or early stage drugs, it's a completely different ballgame, and it really it necessitates that we retool our company engagement strategy and how we interact with companies. So one of the things we did this year is we kind of wrote this company interacting modes playbook. So we have this new playbook that dictates kind of how we interact with companies and try to grease the skids. And most of all, it requires us to be catalytic and opportunistic. All right? So ask any drug developer what their priority is, and they'll tell you it's the patients, right? And I have no doubt that that is true. But in reality, it's not quite that simple, right? It's typically a critical mass of patients whose collective cases can lead to billions of dollars a year in revenue, right? And so in order for companies to become interested in chordoma as a cancer type that they want to develop their drug in, we have to be creative, we have to be scrappy, we have to find ways to be catalytic and take advantage of opportunities when they arise. And so that's what I mean when I say that we've completely changed the playbook for how we engage with companies, because with an ultra rare disease, you absolutely have to take advantage of those opportunities. And so what we've found is that there are these windows of opportunity that exist when companies might be interested or incentivized to invest in chordoma. And so what do I mean by that? So here's a couple of examples. So one opportunity might be that a drug is in phase 1 clinical trials. And what's unique about phase 1 clinical trials is that they're focused on safety, not efficacy, not how good does this drug work. That's kind of secondary. They're most interested in assessing the safety of the drug, and they want to complete that trial as quickly as possible. So they typically enroll patients from all solid tumor types. So there's an opportunity there to sneak chordoma patients on phase 1 trials. And so in the lab, we have to generate preclinical data to justify the enrollment of those patients on that trial. But if we do, then we can get chordoma patients on that phase 1 trial. And if there's activity, then the company might say, hey, wow, maybe we should develop our drug in chordoma if this drug is working really well. So that's one way that there can be an opportunity that we can take advantage of. And it requires us to get the right data into the hands of the right people at the right time. Another opportunity might be an AI drug discovery company that raises a ton of money to expand their pipeline. So now they have this money, they have an opportunity to expand their therapeutic pipeline. So the onus is on us to de-risk brachyuria as a target, make it attractive for that company, and to incentivize them to give it a shot. So we kind of think of this, it's almost analogous to big wave surfing, where we have these windows or waves of opportunity that are arising, and that wave crests at a certain time, and we want to make sure that we get that surfer on the wave. And so CF Labs is kind of a jet ski that needs to tow in a chordoma surfer and put them on that wave of opportunity at exactly the right time. So this is what I mean when I say that the game has changed. We have to be opportunistic because companies are much more kind of protective of their drugs when we're talking about experimental drugs and early stage drugs in development. So let me give you an example of a wave that we're riding in experimental drug repurposing right now. So several years ago, we funded a project in Stuart Schreiber's lab at the Broad Institute of MIT and Harvard, and he used Nobel Prize winning technology that some of you may have heard of called CRISPR to systematically delete all of the genes in chordoma cell lines and ask which one of those genes is essential for the survival of chordoma cells. One of those genes was brachyuria. Another one, interestingly enough, was called SHP2. And this was interesting to us because at that time, there were multiple SHP2 inhibitors in phase 1 trials. I think it was somewhere between 7 and 10 companies had SHP2 inhibitors in phase 1 trials at that time. So we rapidly moved in the lab to acquire a SHP2 inhibitor and to test it in mouse models of chordoma. And this is, I'm showing a couple of examples right here. This is essentially tumor size on the y-axis and time on the x-axis. Mice that are untreated control mice are the black line, and then mice that received a SHP2 inhibitor is the blue line. So you can see that the SHP2 inhibitor significantly slowed the growth of the tumors in mice-bearing chordoma tumors. So we generated a preclinical data package as fast as we could, shared the data with our medical advisory board. Collectively, they all got together, reviewed the data, said, okay, this looks great, and they started enrolling chordoma patients on phase 1 trials for SHP2 inhibitors. And patients started benefiting, including Todd, who was an avid bicyclist before sacral chordoma. The progression of his sacral chordoma led to decreases in his quality of life. He was unable to bicycle anymore, and he was enrolled on the SHP2 inhibitor trial, benefited from it, was able to get back on his bike, and enjoy doing the things that he loved. He came down last summer to the lab in Durham and did a little piece on our local news station about this story with us. And so Todd is doing well, but more patients benefited as well. And ultimately what happened was 2 companies saw that chordoma patients were benefiting on their phase 1 trial. And they said, what's chordoma? Let me learn more about chordoma. And also, let's enroll more chordoma patients on our trial. One of those companies, the data is now public, so I can share it. It was presented at the world's largest cancer treatment conference earlier this summer called ASCO by Dr. Mernal Gounder. So this company, Erasca, ended up enrolling, they expanded their trial specifically for chordoma patients, and they ended up enrolling 13 patients on their SHP2 inhibitor plus cetuximab phase 1 trial. And amazingly, this is a plot that shows the change in tumor size, and this is 0 right here. 11 out of 13 patients on that trial experienced tumor shrinkage. And also these patients experienced long-term disease control, and that was associated with significant quality of life improvements as well.
So is this a combination treatment or just a single?
Patients in red, the red bars, received the combination of what was eventually ended up being the recommended phase 2 dose of 40 milligrams BID SHP2 inhibitor, ERAS-601, plus cetuximab. The blue and the teal bars was single agent. So they had the monotherapy, and then they also had kind of a combination escalation arm too. So it was a little bit of a mixed bag.
Looks like the combination works better.
Looks like the combination works better, but there was only 2 patients that got the monotherapy. 1 patient had a tumor grow and 1 patient had a tumor shrink. And the patient, interestingly, that had a tumor shrink received a lower dose, but actually this was BID, so this was twice daily, lower dose, and then this is TID. So a little bit hard to say, small sample size.
Yeah.
But so that's actually leads me to my last point, which is that unfortunately Erasca halted development of their SHP2 inhibitor, as have most companies in the space right now, for a variety of reasons, but they have still agreed to supply drug for a phase 2 clinical trial with just the monotherapy, the SHP2 inhibitor alone. Because that's a question, that's an open question that we have is what's driving the efficacy? To what extent is it the combination versus cetuximab alone versus SHP2 inhibitor alone? So we are preparing to launch a phase 2 trial in New York City in early 2025 with just their SHP2 inhibitor. It'll be a smaller trial, 10 patients, I believe, and we'll assess the activity of the monotherapy.
Can you use another SHP2 inhibitor instead of this one?
Well, yes. Right now, so the key is, there are a few key ingredients, right, to getting a clinical trial. One, you need a doctor who can run, in a site who can run the clinical trial. We have that check. The second is funding. Thankfully, we have that check. The third, and the most difficult often, is access to the drug. And that's what requires this unique playbook. We have to get the company to become interested enough in chordoma to supply the drug for the clinical trial, and potentially even offer some funding support for the clinical trial as well. So Erasca is...
Any SHP2 inhibitor approved by FDA?
No, they were all in phase 1 trials, and had just many of them, they're at sort of different stages along the way, but some of them I think started phase 2, but almost uniformly companies have abandoned development. But that means that there are out-licensing opportunities. So kind of the play here is we're gonna see how the trial data looks, and then based on where the development landscape is at that time, we're going to explore opportunities to continue the development if the data look good.
So when they stop the development, is it because lack of efficacy or something else?
So they don't make that information public, so we can only speculate. And I think most companies were developing their SHP2 inhibitors in RAS-driven epithelial cancer, so like lung cancer, colorectal cancer, pancreatic cancer, that are driven by the RAS pathway and certain upstream receptor pathways. And SHP2 is just not a genetic essentiality in those cancer types. And even in combination with other drugs, it wasn't working that well, I think. Part of that's also competition in those markets. So the development of RAS inhibitors, I think, has kind of played a role. And so it's like there's business calculus as well. But this is all just speculation. Yes, Sue?
So Dan, if I understood what you said, in those other cancers, it wasn't probably as great a results, but for chordoma patients, it looks good. Is that what happened with the drugs? We didn't have enough to make.
Yeah, so the preclinical data suggests that it's a selective genetic essentiality in chordoma, and that the key word is selective, not just a genetic essentiality in chordoma, but selective. So in the Schreiber paper, he compared the essentiality of the gene in chordoma to that in every other cancer cell line that exists, so across all different cancer types. And in chordoma, it was more selective than, significantly more selective than most other cancer types. And so that's what the preclinical data say. The clinical data, some of it is published, some of it is not. So it's just speculation. And so I am guessing that the activity coupled with the sort of clinical development landscape and market opportunity kind of, that's the calculus that they did. But it's been the past 2 years have seen a lot of changes in drug development. A lot of people are whittling down their pipelines because there's been less investment activity across the board. There has been a lot of investment activity in certain areas, but the access to capital has been an issue, and so a lot of companies have been narrowing their pipelines. And I think SHP2 inhibitors, because of the competition and perhaps because of the lack of ideal efficacy in other tumor types, the development was halted. But again, this is just speculation.
Is that the side effect profile for this?
That's a good question. So specifically from this trial, it was a bit difficult to deconvolute the side effect profile of the SHP2 inhibitor from that of cetuximab. They are kind of in the same pathway. So I am guessing that it would be kind of skin, like rashes and GI toxicity. But again, in this trial, which is published, it was a little bit difficult to deconvolute the contribution from each drug. But it was manageable. The combination was tolerated. It was well tolerated.
What do you mean selective in the context?
You're asking about the selective genetic essentiality? Is that right?
Selective, SHP2. You're talking about SHP2 is selective in chordoma? What do you mean?
I think you're asking about the selective genetic essentiality comment.
Yeah.
Yeah. So actually, I don't have a figure here, but so they used CRISPR to delete all of the genes in chordoma cells and ask how essential is that gene for the proliferation of chordoma cells, right? So that's the essentiality part, right?
Those are deleted in like chordoma cell lines, right?
Yes.
Okay.
Yep. So imagine sort of like two axes, right? So one axis is essentiality. So SHP2 is it's an essential gene in chordoma. The other question is it a selectively essential gene? And so they compared the how essential SHP2 was in chordoma cell lines to how essential it was in all of the other cancer cell lines that they had, which was something like 700. I think it was a huge number across various different kinds of cancer. And SHP2 was a selective genetic essentiality in that it was essential in chordoma, more essential in chordoma than in a bunch of other cancer types. Does that make sense?
When they develop the SHP2 in other
They didn't have the Chordoma data yet. Yeah, they had already started preclinical development of SHP2 inhibitors has been ongoing for a long time.
Yeah, I know, yeah.
And this data just came out in 2022, basically, for Chordoma. Yeah. So that's why we hurried so fast to catch that window of opportunity when we were in phase 1 trials and try to share that. So we shared that data with basically every company in the SHP2 inhibitor development space. And then also, while we were sharing the data, our medical advisory board members were enrolling Chordoma patients on phase 1 trials. And so through those kind of parallel efforts, we were able to drum up some interest in Chordoma. Any more questions on that before I move on? Okay, so finally I'm going to provide an example of some waves that we're riding in new drug development. And this is primarily related to brachiuria. So that is our main focus, is developing new drugs for brachiuria. And along those lines, one of the things that's gotten us really excited recently is AI. And there's this article in the New York Times that came out over the summer about how AI is revolutionizing drug development. And there are now over 800 companies, AI companies, that exist and are sort of in various different aspects of drug discovery and development. There's a ton of investment activity, there's a ton of deal-making activity, so lots of investment in AI right now and how it might be able to unlock new opportunities in drug development. So this is an opportunity wave, right? We have a wave here, so how can we take advantage of that wave? AI-enabled drug discovery. One of the things that excites us most about that is that AI has the potential to make undruggable targets druggable. So what do I mean by druggable and undruggable? So this is a druggable target, for example, EGFR. And druggable targets have these deep, easily accessible pockets that drugs can fit into that are functionally important. So all you need to do is develop a chemical compound, a drug, that can fit in that pocket like a hand fits in a glove, essentially, and block the function of that protein. So that's how EGFR inhibitors like afatinib work. Undruggable targets, like brachiuria, have very shallow or no drug binding pockets in functionally important areas. So prior to now, essentially, it has been very difficult, or some call it impossible, to drug targets like brachiuria. But AI is allowing us to change that. And so AI can rapidly screen billions of compounds in a matter of days or weeks, compared to thousands with traditional kind of chemistry screening libraries. And so AI also can complement other developing technologies like protein degradation. And so it really works well with undruggable targets because it can screen a much larger chemical space, and it can also be linked to other technologies that don't require a very deep pocket on your target in a functionally important area. So essentially what AI does is it shifts the bottleneck from chemistry to biology. And so progress now is limited by the existence of relevant biological expertise and capabilities. Most AI companies lack biology capabilities. They're run by chemists and computational biologists. They're not run by translational researchers, and that makes partnering critical if you're an AI company. And so at CF Labs, what we did, and this is what Tyler presented at CTOS this week, is we developed a biological assay pipeline for targeting brachiuria. We have laid the groundwork. Essentially, if you build it, they will come. We've developed all of the assays, the biological assays that are required to target brachiuria, so that the other piece of the puzzle is just AI, an AI company. And so I'll give you an example. So look, we are partnering with multiple companies. We're in discussions with multiple companies right now. All of it, as you can imagine, is confidential, but I'll give you one example, one high-level example of an early-stage AI company, biotech company that we have worked with to try to drug brachiuria. So we partnered up with this AI company, and they performed a screen of 2 billion compounds in a matter of weeks and identified multiple promising hits, so multiple promising chemicals that are binding to brachiuria, that could be precursors of drugs. And those were subsequently validated in the lab. Just by comparison, using kind of traditional or conventional chemistry approaches, our prior grantees took over 3 years to generate compounds with the same potency. I don't say that to like to make them sound bad at all. That's just like, that is the truth of how traditional chemistry works, and it really highlights and underscores the promise of AI. This is what AI can unlock for you. So instead of over 3 years, it took a matter of weeks to get to the same point. We at the foundation have hired some medicinal chemistry consultants who then designed about 150 derivatives of these compounds, modifying different parts of the molecule. So if you look at this molecule right here, all of these shaded blobs here, basically adding different bits and bobs onto this molecule to try to change it in ways, and then we would run it through our assays to understand if that is making it better. Is it working better now when we make these changes? And so through these iterative assay and design cycles, we have been optimizing these compounds to make them bind tighter to brachiury and approach the strength of tightness that we need to make it an actual drug. And so we will be exploring, company partnerships for further development if we can get to that point. Okay, so just to close here, investment across the R&D continuum is yielding a growing therapeutic pipeline. And we have come from in a kind of a time when therapeutic development in Chordoma was largely empiric to now where we have the lab. We are generating kind of therapeutic hypotheses that have strong rationale, that kind of allow us to understand how a drug is working in Chordoma, and by extension, which patients are most likely to benefit from that drug in Chordoma. We've gone from a one-size-fits-all approach to now, kind of gearing towards precision oncology or personalized medicine. We've gone from kind of thinking about single agents to now considering combinations and how drugs might work together to enhance the magnitude and extend the duration of response. And we've kind of pivoted from repurposing approved drugs to now repurposing experimental drugs, new drugs, and also discovering and developing completely new drugs altogether against brachiury. And all of this is thanks to you, thanks to the support and the kind of the donations of the Chordoma community, and we are doing our best to drive progress at a pace that matches your sense of urgency. So thank you, and I don't know if we have time, but I'm happy to take any questions if we do.
We definitely do. Thank you, Dan.
So you're saying that if a brachiury drug is found, it won't be used all by itself to kill the cancer. It will be in combination with something else?
That is TBD. That's TBD. Yeah, it sort of, it depends on what we see. I think eventually I could imagine a scenario where almost everything would be a combination. But it's not necessarily though.
Well, as long as I have you, one quick question.
Sure.
A couple of years ago, when the doctors told me my cancer had spread to my lungs, I started reading all the papers, and I became obsessed with brachiury research. And I read a paper that said, in 2 to 5 years, we'll have a brachiury drug. And what is your opinion of that prediction? And I don't remember who made it, but it was in a scientific paper.
Interesting. Excuse me, I'm not aware of that paper or when it was published, so I don't know how many years have elapsed.
It was in January of 2021.
Okay.
So I just wondered your opinion of that. Is
Yeah.
it optimistic?
Well, I guess it depends on what you mean by drug, right? Because a drug that is widely accessible to patients requires clinical development, right? But there's also a drug that is in phase 1 clinical trials and might be available to some patients. I would say that for a brachiury drug, we're still probably in that 2 to 5 year time frame today, if everything works out well, just because of the time that it takes to develop drugs.
We have 300 tissues in the bank. Can you just do like next-gen sequencing and someone can curate the data and find some common features?
Yeah, that's a great question. So yes, we have, so a few things. That has been done. So there have been multiple studies that already have been published on sample sizes around 100. So cohorts of about 100 tumors, give or take like 50. And genomic sequencing, so DNA sequencing, has not been incredibly informative. RNA sequencing has not been incredibly informative. And so what RNA sequencing and or DNA sequencing have not been incredibly informative. I think in some cases for maybe 10 to 15% of patients, maybe Joe has a different experience, but I would venture to guess maybe 10 to 15% of patients, if they get RNA or DNA sequencing through, Boston Gene or Tempus or what have you, there will be a targetable alteration found, an actionable alteration that predicts response to a therapy based on what's known in another tumor type. So that's obviously a minority of patients. So both on the research side and on the sort of precision medicine side, DNA and RNA sequencing have not been incredibly informative. So what have we done as a result? So we have invested over the past several years in multiple multi-omics projects. So genomics is one layer, that's the DNA. Transcriptomics is another layer, that's the RNA, right? You also have methylomics, you have proteomics, you have phosphoproteomics, you have, how is the DNA packaged inside of a tumor? There's all these different layers. It's like a topographical map. And we must look at those different data layers because they are probably going to tell us something. And once we piece that back together into a more integrated view of chordoma biology, I think then we will learn more about the molecular subtypes of chordoma, right? Not just the histological subtypes, not just conventional and poorly differentiated, but the molecular subtypes of chordoma, how they associate with prognosis, so risk of recurrence, for example, and also how they associate with therapeutic sensitivity. That's what we have to move towards that. We already are investing in that. We have multiple grants that we funded that are looking at all of these different data layers in big cohorts and across, skull-based chordoma, across spinal and sacral chordoma, pediatric chordoma, adult chordoma. We're trying to just get as much information on that as we can, because right now we don't know, because it's just not, the fact is chordoma biologically is not like lung cancer, it's not like melanoma. We're not getting the full picture, or at least a significant, significant view of the picture from genomics or transcriptomics alone. It's very heterogeneous. Molecularly, it's very heterogeneous. Yeah.
Awesome. Thank you, Dan.